principal scientist as well as the director. He is working on especially on the biotics just related genes in the Brassica genome. So that is his area of specialization. Moreover, being as a director of the National Institute, he is as part of the many committees related with the regulatory issues of this genetic engineering as well as the genome editing. So welcome Dr. Ramcharan Bhattacharya. The floor is yours. Thank you.
So very good morning, dear trainees. I know it's bombardment of lectures, so many a time you are fatigued by one after another lecture, and again the topic of this lecture is not very vibrant. It's regulatory aspects, and if you don't find much interest while listening, all blame is to be given to your coordinator because he has assigned me this topic. So, but of course, the regulatory aspect is very important because whatever you do hard work in developing some product in research, until and unless you take care of the regulatory aspects, it will not reach the farmers or it will not reach the stakeholders. And particularly for genome editing, since a lot of enthusiasm, a lot of expectations are around, not only in our country but throughout the world, obviously the question is that how would you take them forward? Them means the products which are coming out of the genome editing techniques or genome editing kind of research. So how do you take them forwards to the stakeholders? For agriculture, it is mostly farmers. So I would try to give you a very brief idea that what is the status quo, what is the kind of regulation that are existing in India, and leave it unto you to assess that whether it's a very difficult kind of thing or it's encouraging kind of thing. But once you look into the requirements of regulatory policies, definitely you will understand that it's very, very important to be aware, to develop awareness about the requirements right from the beginning, because there are many things that you got to record right from the beginning of a particular research, and until and unless you know that, there could be a problem. So how do I operate this? This is the one. Okay, so I am not sure whether you have been told about the CRISPR-Cas pathways. How do they create genome editing? Have you been told in any of the lectures? No. Okay. So since I was not sure, so I thought I'll just quickly tell you the few lines on the technique per se, or how it works. So when we say genome editing, that essentially means that your experimental capability to edit a gene or a precise location on the genome, right? And the matter is interesting because of the precision. You can make changes at the DNA level by mutation, by treatment with various mutagen, but here the interesting thing is you can create. So CRISPR-Cas9 mechanism is present in bacteria. It works to safeguard the bacteria from invasion of any other DNA element, mostly from bacteriophages. So what happens whenever there is an attack by any bacteriophage? Bacteria, they preserve a small signature sequence and get it integrated into the bacterial genome, right? So it's like memory. They keep a memory that, okay, I encountered this bacteriophage. So there will be a whole array of these memory stretches in the bacterial genome, which will be there in the bacterial genome, will be perpetuating along with the cell whenever it divides and so on. The advantage is that when the same bacteria encounters the same phage, then it quickly do the recognition from its memory. Okay, from its memory. How it does? This array, which is known as CRISPR array, they are transcribed into different small RNA molecules, right? These RNA molecules, they go and scan the phage genome. They go and scan the phage genome, and if they find homology, any of these RNA molecule will bind there. Understand? So a set of RNA just searching in the phage genome, some RNA molecule is finding homology, it will bind. And that binding, and that RNA molecule has another domain which carries a Cas9 enzyme. So it's just like you are carrying a gunny bag. So this RNA molecule is carrying the Cas9 enzyme and searching the genome of the phage. Wherever it finds homology, it binds and deploy that Cas9 enzyme to degrade that DNA. So that is called double-stranded breakage of the DNA, and thereby it destroys the phage. So it's a immunity kind of thing. So what happens? You see here this CRISPR RNA, they are finding homology through, okay, this is called guide RNA, which actually finds the homology and bind to the, so this is your phage DNA, right? This is your phage DNA. This RNA has got homology, so it has bound, and then this Cas9 enzyme is being carried by this RNA molecule. So this Cas9 enzyme will cleave. You see, it has created cleavage, right? And then what will happen? There will be cleavage, and there will be sort of several changes or destruction of the path. But to our experimental use, how we are making use of this technology? What we are doing, wherever we want to create breakage, we use that sequence and make this guide RNA. Okay, you can synthesize this guide RNA against a particular sequence, and you can direct this guide RNA to go and bind that sequence. That is your target gene where you want to cause mutation. Understand? Suppose you have gene A, you want to mutate this gene, so you take some sequence, synthesize it as a guide RNA, and then along with Cas9 you introduce. That guide RNA will go and bind to that portion of gene A, and Cas9 will cause cleavage. And once the cleavage is caused, it will undertake repair mechanism. The repair mechanism experimentally can be again directed. You can direct the repair mechanism, but in cell what happens? In cell in general, it is repaired by a mechanism which is mentioned as non-homologous end joining, NHEJ, non-homologous end joining. And while doing that, it is likely that mistake may happen and mutation will be caused. And we look for that mutation. That mutation may be anything. It may lead to stop codon or deletion of some portion, anything, any sort of mutation. So this is the way you cause targeted mutation. The other way is when you cause the breakage, you can supply a template. The template is, it is having the homology with the end part, and then whatever sequence you want to introduce, this dark blue part is representing that. So this end, while getting joined, it will follow the same sort of enzymatic machinery that are involved in homologous recombination, homologous crossing over, homologous recombination. Those machinery it will use, and it will take up this piece while repairing. So as a result, what is happening? You have incorporated certain nucleotides that you wanted to be there. So you can do allelic conversion, means you can change some nucleotide into other nucleotide, and thereby you can create a new form of that gene, which is called allelic conversion. You know what are alleles? Different forms of a genome. Different means they are different by difference in few nucleotides sequences. So this is another way. So whatever way you can experimentally use it to introduce changes in the DNA, and therefore it is editing of the gene. Now today our topic is that how do you handle these products. If you quickly recapitulate that what we used to do or what we do in case of transgenic technology, you might have heard BT cotton widely being cultivated or many other transgenics. So what you do in transgenic technology, you can introduce a new gene, and the advantage of transgenic technology is that new gene is nothing but a DNA molecule. So it doesn't matter where from you are taking it. DNA is DNA. Whether it is from bacteria or from fungus or from plant or wherever from it comes, it is a molecule DNA. There is absolutely no difference. And then it allows you to introduce gene from an unrelated source through the technology of plant transformation. That is the advantage of transgenic technology. And while doing that, what we do, the gene that we want to introduce, we clone into plant transformation vectors, and then those plant transformation vectors are introduced into agrobacterium cells. Those agrobacterium cells are brought in contact with the plant cell, where they deliver the DNA, and then the DNA, that gene ultimately through this vector gets integrated into the chromosome of the plant, and it becomes a part of the plant genome. So you have introduced a new gene, you have made a plant, transgenic plant, and you know that several traits can be modified in a very, very important and desirable way by adopting this transgenic technology. So my purpose of telling this is to make you understand that what exactly is different at the product level when it is a transgenic plant or when it is a genome edited plant. So in genome edited plants, a central compliance committee, there will be a committee of expert members. They will come and see whether it is properly done or not, and then after doing all this trial, you send the results to send the experimental data or results to another committee, which is called GEAC, Genetic Engineering Approval Committee, and they are the body which finally allows you to release it to the farmers. Okay. So in BRL trials, what you do, basically you collect tissues for various safety analysis, you do a large scale analysis of the trait for which you have made the transgenic plants and so on, and then you release it. So this is the usual procedure, and initially the draft says that you have to follow the similar procedure even for genome edited organisms. Now very interestingly, in the year 2022, 30th March, after a lot of deliberation consultation with expert members, stakeholders, government issued a memorandum which said that for genome edited products, exemption to some of the rules can be given. Okay. So this 1986 rules, which governs the regulatory procedure for transgenics or GMOs. Now in 2022, government says that some of the rules can be exempted. What are the rules? Rules 7 to 11. 7 to 11, these rules can be exempted, and it said that while developing these genome edited plants, they has to be under containment condition, and then once they are free from any outside DNA or exogenous DNA, then they can be given exemption, and they can be treated as a normal varieties, normal new varieties, and thus it will follow the procedure that has to be followed for releasing any normal
bred varieties. So exemption is given for rule number 7 to 11. Now, accordingly, again DBT they issued a guideline for the safety assessment of genome edited plants, but remember it is only, we are talking about plants. So all these exemption, everything, it is only for plants. They are not talking about any animal tissue or any microorganism. No, that is not yet exempted. Understand? So far it is plants. They say that certain rules are exempted. What are the rules that are exempted? Before that they said that genome edited products can be categorized into 3 different categories. One is SDN1 type, another is SDN2 type, and third one is SDN3 type. The difference is whatever I showed in the possible repair mechanism of DSB. You remember DSB, double standard break? So I showed it could be repaired in 2 ways. So those 2 pathways are considered as SDN1 and SDN2. SDN1 where you do not add anything without using a DNA sequence template, that if some mutation is happening it will be SDN1 type, whereas in SDN2 when you are giving a template. SDN3 is completely different. It is where you use the same technology for introducing a new gene. So this guideline says that exemption is given for SDN1 and SDN2 types because there the changes are not really gross but only creating mutation or allelic variant, and which can otherwise also happen in nature. So therefore they need not to be treated as GMO. They can be exempted from certain rules, provided at the end of the product development the experimenter proves that they are free from any external DNA. They are free from any external DNA. So this rule 7 to 11 of the guideline which governs the GMOs or regulatory policies of GMOs, the 7 to 11 is it talks about how the GMOs needs to be tested before release. So you can see here it is clearly says that what is prohibited until and unless it is approved by the competent body. You can read some of them, not necessarily you read all the lines. So they say no person shall import, export, transport, manufacture these diseases except the approval of the GESE. Then they say that GMOs, all experiment of GMOs will be only allowed inside the laboratories. It cannot be taken out until and unless the permission is granted. Then it says that GESE shall give direction. I mean, all whatever way you handle a GMO for importing, exporting, producing a product, all these things, they has to be prior permission has to be sought from the competent body. It talks about all these rules 7 to 11 is talking about all the prohibition until and unless you get permission from the competent body. So all these prohibitions are exempted if in case of genome edited plants. That is the advantage. And not only that, they also say the DBT also issued a clear-cut guideline, as I showed, a guideline for safety assessment of genome edited plants. So the question is that 2 things. Number 1, the exemption you can get provided you prove that the final product is free from exogenous DNA. Now where from exogenous, question of exogenous DNA comes? Because to mediate or to carry out that CRISPR-Cas9 mediated genome editing, you have to introduce those things like a gene which codes for Cas9 enzyme, a RNA molecule which is the CRISPR RNA molecule which will do the genome editing in targeted manner. So all these are exogenous elements. But once the mutation is caused, you have to remove them. You are allowed to carry forward only the mutated mutation, whatever you have introduced for doing the mutation, they have to be removed. Okay, how will you remove? It is very simple. You go forward growing them generation after generation, there will be segregation, and you can select a plant where all those things are not there, only the mutation is there. That is the simple way. So experimenter has to prove there is no exogenous element of DNA, then you are eligible to take those exemption, and whatever plant you made, it will enter into the same procedure that is followed for normally or traditionally bred varieties. Clear? But it is all these things exemption we are talking about only for plants. Understand? Only for plants, no other organism. So then the guideline provided a complete guidance that how to deal with this kind of experimentation right from the initiation of the research. They say that there is all the institutes, they have to develop a institutional biosafety committee, and when you start the research on genome editing, you furnish all the details as per a specified format to the institutional biosafety committee. They will look into these things and they will grant you permission to start the research. And then time to time you have to update them about the development, whether you have developed T0 plants, whether you have got a mutant plant, whatever you are achieving, time to time you have to update IBSC. Now how IBSC will function, all those guidelines are again clearly provided by DBT, so that all institute can have an IBSC and it can function with the proper mandate. And then you see here all those things are mentioned, that investigators are recommended to provide the following information to the IBSC: number of T0 transformant generated, the generation in which mutation was detected, whether that generation of the mutation was in homozygous to the IBSC. And then IBSC regularly monitor the activities as per the handbook for IBSC and extend guidelines. Now the question is that when you are done with the development of the plant, now your job is to prove that they are free from exogenous DNA. How will you do that? So there also a clear protocol is provided, a clear guideline is provided, that how will you provide how will you generate evidence to confirm that genome edited plant is free from exogenous DNA. What you have to do, just let me first come, I will come back. So how will you prove that your plant is free from exogenous DNA? The first thing they say that you have to demonstrate the absence of selectable selection or scorable marker. You know, while developing the transgenic plant, usually one has to use a selectable marker like kanamycin resistance, hygromycin resistance. There are several types. So you have to show that selectable marker is not there, absence. Okay. Second thing is overlapping PCR or nested PCR. Whatever you have used for creating mutation, you have to do overlapping PCR or nested PCR for all that portion, tDNA portion, all that portion, and so that PCR amplification is not coming, and thereby you claim that it is not there in the plant. Now plant is free from all these elements. So you are looking for absence of PCR amplification, and that PCR has to be done in an overlapping manner. Means suppose this entire length you have to check, so you develop primer for this, again overlapping manner, another primer, overlapping manner, another primer, like that. So, and then you show that amplification is not happening for any portion, so you say now exogenous DNA is not there. And then the overlapping PCR has to be done in such a way that the maximum length of amplicon will be 500 base pair, and overlapping has to be minimum 50 base pair overlapping. And then you have to take all sorts of possible control to prove that you are not doing any mistake in reaction. Sometime you may not get amplification, but that is because you have done some mistake in the reaction. So that is not allowed. So you have to take proper control to prove that you are not doing any mistake in the reaction, and thus you will show the absence of exogenous DNA by PCR. Okay. Now if you look into the entire regulatory requirement while doing this research, the first step is submission of application to Institutional Biosafety Committee, which will be reviewed by IBSC and then confirm the proposed category, whether it is SDN 1 type or 2 type or 3 type. They will confirm that, and then all activities in the development process, all activities has to be in the contained facilities, means inside the laboratories or some glass house facility under contained condition, and then final report has to be submitted as per annexure 1 and 2 IBSC, proving the absence of
all those PCR data, assay for selectable marker, all those data you have to provide to the IBSC, saying that plant is now free from exogenous DNA. IBSC will go through that, and then finally they will give you approval that okay, it is certified that your plant is free from exogenous DNA, you can go. So you are not requiring further permission from PSC or any other body. IBSC can give you clearance that okay, and then what will happen? They will enter into the right and track. Right and track, you know that whatever is developed through plant feeding, those varieties they have to go through all India trial. Then if they perform well, then they are released to the trial. So just not once it is proven that it is free from exogenous DNA, they will join the same state of protocols which are being followed for the use of trial. Okay. This is all about how you run the PCR. It's all available in the website. So the complete guideline is available in the website. If any of you is working in slides with your interested, you can do that. And then it also says that one can use any other method to prove absence of exogenous DNA, and that is also okay. That is also okay with the IBSC, provided your method is convincing. Okay. And then to keep a uniformity of genome-edited products, this guideline also suggested a way to name the genome-edited product. A specific way that how do you name the genome-edited product. Number one. For example, for a mutation of 60, CISAR array accessor number this, USC number this, D1 form number this, defect number 1, the name will be, so first two letter will be the genus name, EI, the next two letter will be then it will be the name of the accessor number that you have used to create the mutation, parent line, accessor number, and then there will be the D1 form. D1 means where you submit to IBS for seeking submission or giving the details. That is D1. So reference of the D1 form, what you submitted, and then event number. Event number means when you do the experiment, you may get 5 different each of them is different events, and in each of them mutation would be different. So accordingly, it will be different number. So this is the specific way to do the nomenclature of any products or plants. The purpose is when DVD do this everything, they can see the decay. What is the plant and these SOP will be reviewed periodically by the ICAR and DVD in line with the technological advancement, particularly the method to be used to that. So they will time and again review it, and if there is some advancement happens, they will configure that and will be modified, and this device will be, if there is any revision, it will be immediately posted to the website or other work. So this is the kind of guideline that one has to follow while working in genome editing, and this is very clear now for plants, and we are still waiting for other still final version that is not come. If it is working on it. So with this I stop, and if you have some time to ask any questions you want.
हेलो
The
हाँ जी साईं राम मैम
major point is
नहीं अभी तो नहीं मैम अभी मैं
since
कहीं बाहर
in
अभी कोई
genome
मीटिंग
editing
में
we
हूँ
are
अभी
not adding any new gene, so there is a scope
और
that once
मॉर्निंग
you
में
prove
सुबह
that there is no external DNA present,
हाँ जी
it will be treated as a normally present. That is the whole point
ठीक
which is made in this
ओके
slide. Okay. Thank you very much. If you don't have any questions, otherwise also towards the development system of device, how much is taken? See, you are making changes at the DNA level. So why you expect all this? It's just like mutation. It's mutation. You know that mutation breeding is happening over last more than 100 years. Yeah. The only advantage is here you are causing mutation in a very precise way. Otherwise, what is the way of doing mutation? Suppose I want to increase gene containing rice, and I found that I do not have a plant which can be used as a donor or which is having a high gene content, can be crossed and brought in. So what we do, we try to create a new variability. And in mutation breeding, then you take a parent variety and will give the treatment with mutation. And then I found that I do not have a plant at random several changes will happen in the same donor. Whether it is chemical mutation or physical mutation, whatever it is, several things. And then you grow them, you try to identify a mutant which is giving the trait that you are looking. That is the mutation. Problem is what? Problem is in doing this kind of work, you have to grow a large number of plants. And then you have to very critically observe and find out the plant that you want. There is no guarantee that in that plant, no other collateral changes which are undesirable will not be. Because you have treated the whole DNA, so there could be other mutations which are not desirable. So again, you enter into a crossing program to separate them in segregating population. Separate means your desirable mutation you are separating out from undesirable. So again, several years of. So the advantage of this technique, you are doing the same thing, but you are doing as a precise location. So once the changes at the DNA level happen, there is logically there is no way and to suspect that they would be because they are just a mutant. Now, this level of verification, I think, but knowing the regulation is more important. Everybody working in the country. So, the way things are coming, regulation, this is safety regulation. There is another regulation which is ICR, Intellectual Property Right. Okay. So things are becoming uh, challenging, and as a student, you must understand or you must follow that how things are becoming complex, and that will make you feel more responsible to keep records of your because I mean, what I observe in students that I hardly see any student who has a habit of writing in detail about the experiment that he or she has done. That. And you never know how that record will be very much essential to justify your work of many years. Maybe आपने 10 साल से कुछ बनाया और उसका आइडिया आपको नहीं दिया जा रहा है because they are asking you a document which you have not kept. Hypothetical example. मॉलिक्यूल ए आपने भी डिस्कवर किया मैंने भी डिस्कवर किया दोनों का पेटेंट एप्लीकेशन किया अब क्वेश्चन आता है कि आपने एप्लीकेशन तो 2024 में डाला किसने पहले डिस्कवर आप बोलोगे मुझे तो ये 2018 में दिखा था बट आई टूक टाइम फॉर थॉरो कैलकुलेशन बिफोर सबमिशन ऑफ द The other guy, maybe he has started getting it. वो 2023 में काम शुरू करके तब जाके देखा और किया हो सकता है आपका एक पोस्टर से कुछ क्यों मिल गया था उन्होंने उसके आधार पे कर दिया ये वजह से मैं तो 2015 से ही तो हो सकता है जनरली आपको पूछेगा कि आप लैब रिपोर्ट ला सकते हैं ऐसा मत सोचिए कि उस दिन हमारा करना no they have way to make sure it is genuine तो वही है जितना छोटा से छोटा काम भी कर रहे हैं उसमें रिकॉर्ड वो आता है मैं assume कर रहा हूँ कि 90% आप लोगों में नहीं क्यों नहीं होगा because of good presence you feel it is much better idea which is not directly you have the passage or it is new posting rather than spending some time to write that's very so there is a drive to make us work so we have to resist them drive is from whom drive is from those millionaires जिनको इससे फायदा जो व्हाट्सएप चला रहा है वो चाहता है व्हाट्सएप भी लगे क्योंकि उससे उसका अर्निंग होगी क्या आपको जो फेसबुक चला रहा है वो सोचता है कि आप सारे पब्लिसिटी फेसबुक में ही तो क्या होता है पूरे टेक पेपर का होता है he or she doesn't spend time to think about the next paper but वो substantial time spend करते हैं कहाँ पोस्ट करते हैं ये पेपर आया मेरा ये पेपर आया मेरा ये पेपर आया व्हाट्सएप पे फेसबुक में ट्विटर में आउट चेकिंग तो आपको सोचना है कि टाइम तो 24 घंटे है आपको सोचना है कि आप what for yourself or you will work for others who are making profit out of you. Thank you. Thank you, Dr. Ramcharan. A small answer from our organization. Thank you.
Thank you, sir.
Okay, all the best. Each one of you. Otherwise, also you can contact us for any clarification and any question.
Uh,
Quick break.